Yes.
Yes.
So great. So we just got a pizza come in, but at the meantime, maybe I can just like try to introduce myself and introduce Dr. Caroline Uhler. So she's a core institute member of the Broad Institute of MIT in Harvard, where she directs the Eric and Wendy Schmidt Center and is a member of the scientific leadership team. She's also a professor of engineering in the Department of Electrical Engineering and Computer Science and the Institute for Data System and Society at MIT. She's research lies at the intersection of machine learning, statistics, and genomics, with a particular focus on causal inference, representation learning, and gene regulation. And she receives multiple awards from NIH, some Simons, and NSF. And she holds a master in mathematics and a bachelor of science in biology and the MED, all from the University of Zurich. She obtains her PhD in stats from UCB and then spent 3 years as an assistant professor at IST Australia before joining the faculty of MIT. So today she will be giving a talk on multimodal data integration. Welcome everyone for her.
Great. Welcome, Caroline. Thank you for being here.
Thank you.
And thank you very much for having me. Okay, so you guys enjoy your pizza. Would love to have a piece as well. So I will talk about multimodal data integration and in particular how we can maybe use multimodal data in order to go not just to new biomarkers, but actually all the way to causality or mechanisms. Now, of course, we all know, multimodal data is all over the place in the biomedical field, be it at the cellular level, where we have, RNA-seq, single-cell RNA-seq, all kinds of imaging modalities, at the tissue level, at the organ level, say here, heart MRIs, heart ECGs, heart angiograms, genetics, of course, DNA, and then at the whole organism level, where you can do all kinds of measurements, be it using a smartwatch or be it using imaging. And so it's pretty clear that, we do need some ways of integrating very different data modalities and also at different scales in order to get a better picture of, an integrated picture of the system itself, be it if you're studying tissues or cells or organisms. And if you can do this, if there is a way of integrating very different modalities of data and also scales of data, well, then an immediate opportunity is, of course, that you could then maybe get more comprehensive biomarkers of a particular disease, better characterizations of disease as well. And what I also, and I'll talk a little bit about this, but what I also in particular want to get to is that, maybe multimodality could also help us to get at mechanisms and causality. And that's maybe not so straightforward, but the main intuition is the following, because I will be using this throughout the talk. And by now, finally, we also have some proofs that this actually really holds through. But, if you're thinking about trying to understand what is actually causal in the system, or mechanistic, or let's use the word causal because I will actually talk about causality. Causal in the system, well, different modalities can help you because whatever is causal is more likely, it should be there, right? No matter whether you're looking at it from the top or from the bottom or from the left or from the right. And so, these different views can also be seen as different modalities. So how can we get to causality? It's like, something that is causal in the system should be there, whether you're looking at it from a sequencing lens, from an imaging lens, from a genetics lens, etc. Right? So different modalities might actually help you to get at the causal information because you can then look for whatever is invariant to how you're looking at the system. And so I'll be using this kind of point of view, and that has actually brought us quite far, or this hypothesis has brought us quite far. And we're also more recently actually able to prove that this really also holds mathematically. So multimodality can actually help us to really when we want to get at mechanisms and causality. And so I'll want to talk about this. And then of course, there is also the question of experimental design, because if you can get different modalities, well, maybe some modalities contain more information than others. but maybe they're more expensive, right? And so then it depends on the downstream question whether you actually need a modality because the information you're looking for is only contained in that particular modality, or maybe, you know, it is also contained in a cheaper modality, which you can then translate into this other modality, based on which you can then do the downstream task. But that, of course, only works, this translation task. Well, you cannot generate information that is not contained in a modality. Say if we're going from single-cell RNA-seq to single-cell, say, DAPI, right? If there is some information, or the other way around, because DAPI is cheaper than single-cell RNA-seq, well, if you can translate from single-cell DAPI to single-cell RNA-seq, well, there might be some information that is actually not contained in DAPI about RNA-seq. Well, so then if you translate, for sure, you cannot just generate it. So that information will just be lost. And so if that's exactly the information you need for the downstream task, then, you know, then you should better get your single-cell RNA-seq data. So there are important experimental design questions about what information is contained in a modality that is not contained in another one, or what information is actually shared between two very different modalities. And then, of course, the cheaper modality would be sufficient. So those are the types of questions that I want to get to, which, you know, is maybe a step further from actually just looking at biomarkers by integrating many different modalities together. And please feel free to interrupt me. I don't know if I will hear it from the room, but maybe someone then could just stand up and actually talk into the laptop. Okay, so why do we need causality? I think that's probably clear for all of us here. I just have a very simple example. Fibrosis, an example where we don't have any drugs yet that can revert fibrosis. And so, you know, in order to identify new or potential drug targets, it is really critical in order that you are able to identify genes or proteins that cause tissue stiffening, and of course differentiate that from genes or proteins that are affected by tissue stiffening. So you really need to know what is upstream and what is downstream, because only what is upstream is a potential drug target, and whatever is downstream, there will be a lot of genes and proteins that are associated and affected by tissue stiffening, but those are not good potential drug targets. However, you know, if we look at the current, you know, super powerful machine learning models, large language models, generative models, you know, they are actually, I mean, they were never built to take into account causality, and they actually generally fail miserably on causal tasks. So here is a very small example. We all know that, you know, good weather is the cause of higher ice cream consumption, and it's not the other way around. So if you take a gloomy day and you give out a lot of free ice cream, that's not going to change the weather. However, if you tell, you know, ChatGPT to generate an image where, you know, there is an ice cream stand where they're giving out ice cream for free, and you should all try it, it actually always puts good weather in the background. Although you're saying that you're intervening on ice cream consumption, so that really shouldn't change the distribution of weather. In particular, you know, if you just ask it to draw a park, and you will look at the weather, you know, there is a certain distribution of seeing rain and sunshine and snow, etc. Whereas as soon as you say something about ice creams, even though you're telling the model that you're actually giving out ice creams for free, it is just it's conditioning instead of intervening, right? So if you condition on ice cream consumption, that actually changes the distribution of weather. But if you're just intervening on ice cream consumption on something downstream, you're giving out free ice cream, that doesn't change the weather distribution. And so, but, you know, current AI models were never designed to differentiate between cause and effect. And so it's critical, but for biomedical applications, of course, this is the critical task. And so we do need new models that can actually take into account causality. And so that's a lot of what we do in the lab, really thinking about how can we get models that, you know, scale and go towards, you know, these important applications that we all have in mind, but then actually are causal as well. Okay, so how do we think about causality? Well, you know, in the biological sphere, I guess, you know, the gold standard would be, oh, we would love to be able to draw these full regulatory networks. Here, for example, a gene regulatory network by this, you know, beautiful work by Davidson on the sea urchin, where literally in like very, very, you know, a lot of experiments, it was actually figured out, you know, which gene or it regulates which other one, and even differential equations on all of these directed edges. And so these kinds of networks or directed networks can really be seen as, you know, as a causal network. Sewell Wright developed the foundations for causal inference already very early on, actually by studying heredity. And so statistical framework was set up, again, coming out of more biological types of questions, which is interesting. And so these directed edges here really represent causal relationships, meaning if you change here one of these variables like G prime, then H prime will change. And then, these edge weights here tell you by how much it will change. So, often this is seen as the gold standard that we would like to actually learn these full regulatory networks. Now, we've done a lot of work showing that, on like 20,000 nodes, we simply will never have enough data to actually learn regulatory networks on 20,000 nodes. So I'm not going to talk about, really how to learn these very large scale networks just from data itself. I just don't think it's really ever going to be possible to do. I mean, we just have mathematical proofs that that's, you'll just never have the types of data sets that you will ever need in order to learn these causal networks. But also it seems that maybe this is not, or I guess in the biological space, we also have another question, namely, generally, we don't even know what the nodes are. So, say I have, and we nowadays have a lot of imaging data where we actually have perturbations, like we have drugs supplied to cells, we have genetic perturbations applied to cells, and then, we have as in a readout, say, imaging data just because it is the cheapest. Now, it's not clear in an image like this of a single cell, here's a nucleus image, what are the nodes? What are the nodes of this the causal graph, right? It's certainly not pixels. So because this pixel here has nothing to do with the same pixel in a different image, because, right, these cells are just put there in some way. So there is no alignment of cells. And, the causal features would have to be learned, right? It could be, for some problems, it could be maybe the shape of the nucleus. It could be how much of a particular protein is inside the nucleus. It could be where this particular protein is inside the cell, etc. So the causal variables themselves actually have to be learned. We don't know them. And then, once you learn the causal variables, then you would have to learn these networks. But maybe the causal variables are not that many, so that we can actually learn these networks. So maybe we shouldn't start by, learning a network or trying to even learn a network on 20,000 nodes just because these are the protein coding genes. Because, even if you have single cell RNA-seq data, it's not clear that every causal variable is just one of the genes. It could be, right, that many genes work as protein complexes, and so it doesn't really matter which one of them you're perturbing. So they should all be together one node, one causal variable. And so even in the setting where we believe that we know what the causal variables are, I think it's actually not clear that we really know them. We really, even in that setting, have to learn them, and there might be much less causal variables than our 20,000, protein coding genes. And so the problem is really, how do you learn actually even what these causal variables are? Okay. And so, and now, of course, we do have multimodal data. So we might have imaging data, we might have sequencing data, or, I'll show afterwards, like the same frameworks hold for very different, like if you have heart ECG, heart MRI, heart angiogram, etc. So the same frameworks hold in all kinds of different applications. And so now the question is, how do we learn these variables, right? So I have a lot of these images. How do I even learn the variables that could then afterwards be causal? So we'll argue about causality later on. But first, we just want to find these variables. And so, this is, of course, representation learning, right? So you have a lot of images or a lot of single cell RNA-seq data. This is now cell level. We'll do it at the tissue level, organism level afterwards. RNA-seq data, single cell attack-seq data, whatever data modalities you have. And now you want to learn the variables. So as we said, right, like the pixels here are not variables. Even in single cell RNA-seq, the genes themselves might not be variables. Similarly for attack-seq, it's certainly not that every peak is a variable. So we may also need to learn the variables here. And also in attack-seq, we usually assume that something like you organize the data at the gene level. Well, it's also not clear that you might not have two causal variables for a gene and maybe no causal variables for another gene. So we want to learn what these causal variables are. And so we've developed a lot of work using autoencoders to do this. So, these neural networks that map to some latent space. So this is where our causal features live. And then, they're trained so that they go to a latent space and then back to each one of the modalities, and they're trained through reconstruction loss. And so now when you have multivariate data, so you can do the alignment of these different modalities. And, you know, so now here comes the intuition from causality that, you know, which really tells you somehow they're you know, in many different papers, you see multimodal autoencoders, but often what they would do is like either concatenate the different features. So say you have an image, let's take two modalities that are, say, paired. So we have single-cell RNA-seq and single-cell ATAC-seq. So you go to the latent space and you concatenate the two features of the RNA-seq and ATAC-seq, or you add together the different features of the single-cell RNA-seq and the single-cell ATAC-seq. But really, if you're coming from a causal perspective and you think about, you know, what we want to find, the variables in here, they should be invariant to how you're looking at the system. Really, what you should be doing is actually only learn one set of variables. For sure not concatenate them, for sure not add them up, but, you know, just learn one set of variables where, you know, the RNA-seq, if you have paired data, the paired single-cell RNA-seq and ATAC-seq should be mapping to the same point in the latent space. And that can just be put up with, you know, adding, say, a contrastive loss, where you're making sure that these two data points actually end up close together in the latent space. And similarly, in fact, exactly the same frameworks can be used if you have unpaired data. So we did this, for example, with single-cell images and single-cell RNA-seq. Now you don't have a pairing, so I don't know which cell belongs to which cell in the different modality. However, you can still do it, and I'll show you an example on the next slide where you say take a population of cells. We will be taking T cells, and at random I take out some for imaging, and at random I take out some for sequencing. Now I know it's the same population of cells, so at the distribution level, they should match in the latent space. So there, instead of, you know, matching at the single cell level, we are going to just match at the distribution level, meaning you have, say, a discriminative loss that says, you know, my embedding is a good one if I cannot tell whether a particular cell comes from RNA-seq land or from imaging land. Okay, so you can still align, even in the unpaired setting, you can have these joint distributions in the latent space. And now here I explained it in the cell level, but we will see, you know, exactly the same types of frameworks, of course, can be applied in all kinds of other settings. And, you know, nowadays you can also use, if you have enough data, we often don't have enough data, you can use transformer architectures to go into the latent space. And of course, you know, if you have molecules, say you could use graph neural networks to get into the latent space. So all of these encoders, decoders here, they really, you can, you know, they are based on whichever modality that you're actually working with. So just to show you an example of how this works, so this is the first example, which was actually one of the hardest for us to do. We wanted to take two very different modalities. So this is DAPI stained images of cells, so imaging data, and then single cell RNA-seq, which could never be measured together in the same cell because getting every measurement means killing the cells. And so all of this work, in fact, even also on the last slide, is all in collaboration with Shiva's lab, GB Shivashankar's lab at ETH Zurich, with whom we've been collaborating for over 10 years or so, and really has really built our program as well, the kinds of biological problems that we're actually interested in. And so here is, you know, from very early work in Shiva's lab, they saw in mouse CD4 positive cells, in naive T cells, that they actually found two different populations just based on imaging, some with the heterochromatin, you see it here nicely, in the outside, and then some with the heterochromatin in the inside. And then also showed that these actually, how the DNA is packed actually also matters in terms of functional state. The heterochromatin in the inside, these cells were actually softer, and they showed accelerated activation. So now in collaboration, we got the same data also in humans. So again, CD4 naive T cells. And in fact, again, if you just do clustering of these images, you see again, very clear two clusters. One has heterochromatin more in the outside, and one has more heterochromatin in the inside. Now the question is, can we somehow link this to what is the functional state or the transcriptional state of these cells? So now we go to single-cell RNA-seq. So even in single-cell RNA-seq, if you do clustering of the naive population, you very, very cleanly see two clusters. Here, what we did is we also added the activated T cells, just in addition as a data set, just to show that one of these naive populations is closer to the activated state than the other one. Okay, you see it also here nicely in this t-SNE plot. So we have here the activated state. We have here the green population, which is the one that is closer to the activated state. And then you have the blue one. And so although these are all naive cells, T cells, we just call them, this one here poised for activation and the other one quiescent. Now, the question is, of course, can we link somehow these images to which state they come from? And so now we use exactly this autoencoder model in order to link in the latent space the distributions together. And that, of course, gives you then a mapping from one modality to the other at the single cell level. And so, right, so now we can translate between images. So these are these DAPI-stained images to actually single cell RNA-seq and ask, where do we end up? And in fact, from these, we were predicting that, you know, the ones with the DNA in the inside are the ones that map to the activated T cell state, which was also what was, you know, suggested here with a central heterochromatin state being the softer one, which can be more easily activated, that this might actually be the one that is poised for activation. So that's how you can now connect and translate between very, very different modalities. Something like here, these DAPI-stained images, and then actually single-cell RNA-seq. Now, of course, you can ask, well, some information might have gotten lost, right? But at least at the state level, we were able to translate between these two very, very different data modalities. Now, how well can we translate? So this was just, you know, at the state level, as I said. So nowadays with spatial transcriptomics, we can actually validate these kinds of frameworks, right? These kinds of translation frameworks between very different data modalities. So this was now, again, in collaboration with Shiva's lab, and then also with Xiao Wang at the Broad, Xiao Wang is who developed StarMap. So this, you know, imaging-based spatial transcriptomics modality. And so now it's really nice because now we have paired data. So now we will have, you know, in this case, it's Hoechst, but still it's chromatin staining paired with gene expression data. And so now we can actually directly validate, right, how well, so again, our models, how well they can actually translate between just these chromatin images to actually getting out gene expression data. And so how do you read this? How is this trained? Again, same as before. We have here two modalities. Actually, we have three modalities because we also have the spatial context, which we also used. And so every dot here is a cell, right? And two cells here are close together if they have similar gene expression profiles and they have a similar imaging profile. And then we just do clustering in this latent space, like how you would do it, you know, when this is just a representation using UMaps, same thing as how you would do it when you have single cell RNA-seq. You then do clustering in this latent space, and these are all our clusters. And now you can ask, what do these clusters mean? You can map these clusters back to the imaging space, for example. And these clusters, you know, nicely correspond to different cell types. But in particular, we can then validate what we did before with like asking, well, you know, it seemed like we can translate quite well just from DAPI-stained images to gene expression. And now that's, you know, now we took actually, this was in the mouse brain in an Alzheimer's model. So we took a new slide, so a new brain slide, data we have never seen, right? We have trained it on other mice. Now here comes a new slide. All we're using is just these images. And we translate it based on this trained model. We translate it to gene expression data, so single cell RNA-seq. And now you ask, well, you know, let's do the same analysis as like, you know, Xiao was doing before in her lab on the single cell, on the spatial transcriptomics data. And so based on the generated gene expression data, now you just do clustering on it. You identify the marker genes like your standard single cell RNA-seq pipeline. And we actually see quite nicely that just based on the images themselves, we can identify all of the different cell types that, you know, the major cell types that were used also in the original publication. You actually find out the correct marker genes just based on translating from DAPI staining or Hoechst staining to single cell RNA-seq. So that's as much, you know, information as is actually contained in DAPI or Hoechst. So literally you have enough information to get all the cell types out, to get all the marker genes out, just based on these, you know, very cheap images. Now, of course, you can do more. You can, get your you can use both modalities to get to new biomarkers of disease, in this case, Alzheimer's. As I said, this was an Alzheimer's mouse model. So based on the embedding of each one of the cells here, you can ask, can you predict, based on that cell, can you predict how much plaque there is nearby or whether there is plaque nearby? And that's exactly this prediction task that we trained the model here to do. And all you get here is the gradients. So, what is highlighted here are pixels that are very indicative of having plaque nearby. Whereas when it's dark like this, it means that these pixels are not indicative of having plaque nearby. And so you see something interesting, right? You see some cells, like some microglia here, are lighting up, which is what you would expect, right? Usually people would define this microglia that are close to plaque as disease-associated microglia. And you see here the model picks out some of these, but it actually doesn't pick up other microglia that are also close to plaque. But it picks up even things like inhibitory neurons, etc. So it even picks up neurons as being indicative of being predictive of having plaque and how much plaque nearby. Now, of course, you can stop here. You can say like, okay, so these are biomarkers, but it's better if we have some interpretable biomarkers that are actually simple ones. So that's where collaboration with Shiva's lab, who does a lot of this imaging of nuclei, comes in very strongly, is that when they looked at this data, they suggested immediately that what it's actually picking up is areas that are heterochromatized, so again, densely packed DNA. And once there is a suggestion that what the neural network is using is actually that, well, then that's all we're going to plot, right? So then you can actually plot heterochromatin to euchromatin ratio versus gradients, and you see that that's super well correlated in all cell types. And so then you know that this is what the neural network is using. And well, once you have this, that the neural network is doing it just based on, or, definitely hugely doing it based on something that is highly associated with just, heterochromatin to euchromatin ratio in a cell, well, then let's just plot that, right? So now you have a hypothesis of what it could actually be that is a very simple and, in this case, new biomarker for disease. And it's really quite nice that this actually comes out to be significant in every one of the cell types. So with Alzheimer's, so the DNA compaction goes up, so it becomes more and more compact in all cell types. So that's interesting. That was not known, that there would be even these changes in all these neurons as well. And this is particularly interesting because DNA gets decompacted with aging, right? That's very well known. So with aging, in fact, you have the opposite trend. It's going down. So DNA opens up with aging, but DNA closes off in the disease context. So it's quite interesting to then study which areas are closing off and which areas are opening up in the disease context when you're becoming older. Now, this was all in the mouse. We're very excited. This is a work that is currently, actually, I should say here in review, that in fact, exactly the same trends hold through also in human brains. So here we work together with Queen Square Brain Bank. And, it's very, very interesting that we see exactly the same trends here as well. So, with aging, heterochromatin to euchromatin ratio goes down. This is well known. We saw it again also in this data. But then, with Alzheimer's versus control, heterochromatin to euchromatin ratio goes up. And, now we can look at more about the different cells and which ones actually have this stronger or less strong, and what might be other things that are ongoing, like lamin invaginations, etc. So it's quite nice that there is this new biomarker that is, holds through more generally, and of course also is also something that is interesting from a biological perspective to actually know which areas are closing off more and which ones are opening up more. Okay, so that's, everything modalities at the cell level, tissue level, but, just to show that these same types of problems the same types of approaches, are really general approaches. So here we did it with heart ECG and heart MRI. Again, two modalities where you have paired data in the UK Biobank. You have paired data at the individual level, right? And so again, like before, you have some contrastive loss in the latent space that says, the ECG and the MRI of an individual, if they come from the same individual, should be close together. And of course, other individuals can be far away from each other. And you have a reconstruction loss where, your ECG, and now, of course, now this is a completely different modality. You'll have completely different encoders and decoder architectures than what we had before on the cellular levels. So you'll have a reconstruction loss for ECGs and for MRIs. And then you have this beautiful latent space, which has, one data point, or you can have two for the different modalities, but usually we'll just say one data point for each one of the individuals. And you can start translating from ECG to MRI. We'll ask afterwards, right? Not all of the information will be shared between ECG and MRI. There might be some information that is only in an MRI that is actually not contained in an ECG, similarly as for a single-cell RNA-seq and images, right? But a lot of information will actually be shared between these two modalities. So what we showed also in here or found in here that we can translate quite well from ECGs to MRIs. And read off all kinds of, diseases that are generally, where you generally use an MRI in order to use it for diagnostics. You can actually get it by a translation from the ECG itself, suggesting that information is actually already contained in an ECG. We might just, as humans, we might not be able to read it out of an ECG, but it is already there so that you can translate it to MRI. You can actually read it out there. But then once you have these, integrated representations, you can even start doing things like GWAS in the latent space, where the GWAS is now not just based on a one-dimensional feature, a particular disease-associated feature, like, the size of some chamber here, but actually is, fully, in some sense, unsupervised. So we're just looking at the state of the heart and seeing, what are SNPs that significantly move the distribution in this latent space. What are SNPs that all do similar things? So they're moving the distribution in the same direction, meaning these SNPs all have a similar effect, at least in terms of the readout in ECG and MRI. And you can also decode it. So they are now interpretable. So these SNPs, they will do something in terms of changing the ECG in this way or changing the MRI in that way. So those are the kinds of things, because these models are generative, you can also, of course, always then go back to interpret what directions mean in this latent space. So that's just to show that, these models are very, very general and can be applied in all these different types of contexts. Now, I mentioned, and probably many of you have been wondering, right? So for now, all I'm doing is always going into one latent space. Even from this causal, the causal intuition at the beginning for all of this work was always, well, for now, we'll just be okay with throwing away some information. Namely, we're throwing away all of the information that is not shared between the two modalities, right? Because by making sure that, that whatever you get from an ECG in an individual and MRI in the individuals, you're going to the same spot in the latent space. It means that we're just looking for information that is invariant to how you're looking at the heart itself, whether you're looking at it from ECG land or from MRI land. Now you might say, and rightfully so, well, maybe an MRI has some information that is actually not contained in an ECG, and potentially even the other way around. And so how could we maybe even identify not just the information that is shared between two modalities, but maybe also information that is actually not captured by the other modality, which is also really important to know for experimental design. And so that's something that really kind of was a question that we've been thinking about for a long time, right? So say you have like different protein stains, and we have, our nuclear stain, like DNA stain. How can we figure out, what is information that for which we really need to get this protein stain because it's simply not there in this cheap, DAPI or Herc stain of the chromatin, and which information, is actually shared, meaning I could just take these cheap stains and then just translate to this particular modality. Similarly for ECG, right? So if what you care about is something disease associated that you can actually read out that is contained in the shared space, well, that means that you can just use the ECG, translate to the MRI, and you should be able to read it out based on your MRI, just because that's maybe more interpretable, but you could also get it out of the ECG. And what we always find interesting is when, we didn't actually have an algorithm of how we could do this. How can we identify information that is shared, modality specific? We didn't even know if it is possible. But then actually our theoretical results on, where we're talking about causality and identifying causal features told us in the theorem that, well, we can actually identify even, causal variables that are modality specific. So how you read this is, say we have 3 modalities. Here it's like imaging, some tabular data, some text data. And these Z variables are these latent causal variables, right? These variables that we were learning, for example, through this autoencoder. And we were always only concentrating on this information, the information that is shared between them, right? In this case, Z1, Z2, Z3 would have been learned by these, multimodal autoencoders when we're looking for whatever information is invariant to how we're looking at the data. But in fact, these results told us that we can also identify, say here, Z4 and Z6 and Z7 and Z5. So these are informations or variables that are only contained in this text or are only contained in this tabular data. Or similarly, this would mean that we should be able to identify information that is only contained in this particular protein stain and cannot be gotten by translating from the DNA stain. Now, still, theoretical results usually don't come with an algorithm, but at least it hints at how the algorithm could work. And now we finally also have an algorithm for actually doing this. So this work is actually just going to be, was just accepted. So I should change this as well. So here is an example. So now we have an algorithm that can actually identify what is shared information and what is modality specific. We've applied it to many, many different cellular modalities. We have not yet done it, and something that is ongoing, like to more medical imaging modalities, like our heart ECG, heart MRI, etc., where it would be very interesting. Like say you take a brain MRI and a heart MRI and a heart ECG. Of course, you would expect, although modalities of brain MRI and heart MRI look more similar to each other, you would expect that you have more shared information between a heart ECG and a heart MRI than with a brain MRI. So those are nice use cases to see like how much information is actually shared. So we did it here more in the cellular level, and here is just one particular application for one particular protein stain where we actually thought that this would be a protein stain that we could get from DAPI. So what is this protein stain? So gamma H2AX is a stain for DNA breakpoints, which is something very important, for example, in the cancer context. And we thought because it has something to do with the DNA, maybe, if we just stain the DNA, we should be able to predict where these breakpoints are. And what is particularly relevant, these breakpoints, they're like foci, right? So they're actual breakpoints. So it's always punctuated within the nucleus. And so, you would want to know how many breakpoints there are. And in fact, based on this model, we identified that information, like how many foci you have, is actually only contained in the protein-specific latent space and not in the DNA shared latent space. And then, of course, and there are other things that are contained that you can actually translate. So if you care about things like the texture of these breakpoints, that you can get from the DNA. Okay, but how many breakpoints you have, you can actually not get from the DNA stain. And you can validate that, right? Once you have this based on these multimodal models, you can validate that isn't just, a limitation of our particular model, but then you can train another model to, just input DNA and output, well, how many foci do you have? And you'll see that you can actually not predict it based on the DNA stain, at least in the data sets that we have. Who knows, at some point, maybe you will have enough data to be able to predict that as well. But those are the types of questions that you can now answer, and I think are really important to answer, right? If you have a modality that is much more expensive than the other, you do want to know that it's actually required to get that modality and that you couldn't get it out, the same information out from a cheaper modality. So that's how you use these types of models, these multimodal models, and how you can actually get a causal information even in this latent space. Now, I just wanted to show that, these types of models of trying to translate between modalities, you can take them much further. I mean, it's not just translating between modalities like what we've seen here, but you can also translate to, say, a perturbed state. So in goes a control cell, and out comes the prediction of how that cell looks like in the perturbed state. So for example, a perturbation could be genetic perturbations. Okay, so you have some training data where I have, say, let's look at sequencing data first. So I have single cell RNA-seq data. I have a perturb-seq screen. So I have genetic perturbations as well. And so I can train it for the genetic perturbations that I have. I can train it that in goes some observational cells, right? No perturbation, and I can train it to output perturbed cells. Again, this is in the setting where you actually don't have a paired data, right? Because you never get the cell with a perturbation and without the perturbation. So again, you have to do it, the matching has to be done at the distribution level. But kind of very similar types of frameworks here used using, a transformer so that we also learn the gene regulatory network in the latent space. But really basically same types of ideas of now translating from one modality being no perturbation to the other modalities being different types of perturbations. And, once you have a general framework like this, it doesn't matter whether how you observe the cell is single cell RNA-seq or how you observe the cell is single cell imaging, like in optical pooled screens. You can just apply exactly the same kind of framework in order to go from an image of an unperturbed cell to predicting how the perturbed cell would look like under a particular genetic perturbation, or even under a particular drug perturbation. And now, of course, these kinds of models you can very broadly apply. I think we all know about virtual cell. You can do things like predicting perturbations you haven't seen yet, predicting perturbations that you might have seen in one cell line but not in another one, predicting the effect of combinations of perturbations, do experimental design of which perturbations might be the optimal ones in order to move a cell to a particular state, or then also learn these gene regulatory networks where you learn what the causal variables are, what these modules are, and then you learn the regulatory effects among them. So that's just, to show you how you can now come up, mix and match these approaches for different types of applications. You can go much more functional to the protein state. Similar idea, but now, of course, completely differently put here. So, if you care about proteins and protein localization, which, is arguably a step closer to function than just looking at single cell RNA-seq. Now, it's quite well known, right, that proteins and the localization matters for proteins, so not just the abundance, but actually the localization. Many diseases can actually be brought back or explained by, a particular protein being mislocalized in the cell. And what is also well known is that, a protein can be localized to one organelle in one cell type and actually be localized to another organelle in a different cell type. And even within a cell type, proteins can show different localizations. So they can have variability in terms of where they're localized. Think of a transcription factor, right, which can be sometimes inside the nucleus, sometimes outside the nucleus, sometimes in different places within the nucleus. And so we'll never be able to do experiments where we get to see or get to image every protein in every cell type. There is already a huge resource, which is the Human Protein Atlas. But even this huge resource only measures about 0.25% of all cell line and protein pairs. And so that means, we need to be able to have models that can predict if I give you a particular cell, and now because, right, we have variability even within cells of a cell type, it would be great if I can, give you, say, some landmark stains of a particular cell, say again, DAPI and some microtubule or ER, like some of the stains that are usually available. And I tell you which protein I care about. Can you then predict where this particular protein is localized inside the cell? So again, like a generation type question, a translation type question, I give you these landmark stains, you tell me what you care about, like before it was a perturbation, now it's a particular protein sequence. Can you now generate the image of this particular cell of, like, now you're fluorescently labeling this particular protein. So can you just tell me how the image would look like of that particular protein in this particular cell? So this now, of course, you have some supervision. I mean, you can train this model, right? Because you do have data in the Human Protein Atlas. But of course, it doesn't cover all cell types and it doesn't cover all proteins in all cell types. But, you know, the question is, how well does this work, right? In particular, how well does it work for cell types that you have never seen in training? Maybe even cell types that are not in the Human Protein Atlas. And so this is some validation experiments. So, and this was in collaboration with Fei Chen's lab. And here I mean, these are maybe the results I'm most excited about. You know, in red are all of the cell types that we never saw in training. In particular, there is even one cell type that isn't even contained in a Human Protein Atlas. So it's a cell type that is not there. And even these two cell types we never saw. They are there in the Human Protein Atlas, but we held them out completely from training. This new data, this was actually data that was generated in Fei's lab. And, you know, also similarly here, we have certain proteins that we have never seen in training. In particular, you see some bias. We actually just used alphabetical so that it's clear that, you know, there will be very different protein families. So we just alphabetically chose the first ones all for training, and then all the other ones, we held them all out for validation and for test. And so here you see how well or how poorly or how well it goes. But, you know, I think it's quite nice that you can very easily identify and see differences of, you know, in a true image and in the predicted image, a particular protein that is more localized in the cell nucleus and a protein that is more localized in the outside of the cell nucleus. And, you know, it's also nice when whether you take cell lines that are used in training or unseen cell lines, you actually get equal prediction accuracies here. And so now what do you use this for? Well, you can use it in order to predict protein localizations in particular cells you care about. You can identify, you know, you can answer questions that you couldn't do without models like this, like, you know, what are proteins that are most variable across cell lines in their localization? What are proteins that are most variable within a cell line in their localization? There is no data set to answer that question. You have to generate the data of where these proteins, where you believe the proteins will be localized. And then, you know, you can try to validate some of them, but you'll not be able to validate that in all cell lines. So those are questions where you need generative models, computational generative models in order to answer these questions. But you can do other things which, you know, maybe from a disease perspective is more interesting, right? Because if I go back, I can also ask questions like, well, if I have a mutation in a protein coding sequence and I make this mutation here, well, you can then predict where that protein will be localized, right, with this mutation. And so, you know, then you can look at, well, where is a protein localized in the normal setting and where is it localized with the mutation? And here are just some examples where we did this for some proteins where it's known that if there are mutations or all of these are all mutations that are associated with this particular disease, and that this disease is associated with a protein mislocalization. And so here, although these are the images in the wild type, real images, these are the generated images of how it would look like under the mutation. By eye, you can't really see a difference, but if you actually take the difference in these images, you'll see that, now here there is more protein inside the cell nucleus. So that's this red thing. And you can also ask questions about, well, how did the model make this prediction that now there is more protein in the inside? Which of the regions were important for predicting that it's now in the cell nucleus instead of outside? And here, for example, there would now be a hypothesis that these mutations here at the beginning and at the end are actually the ones that then lead to, the protein being mislocalized, and maybe all these mutations in here are actually not the relevant ones, or not the disease. In this case, the hypothesis would be that these are not the disease causal ones. So that's how you also get to interpretability. You can also do this with, particular proteins where we know where they're localized, but we didn't know why. Which parts of the sequence might actually be the one that means that you're going to be moved to a particular place inside the cell, because you can identify, right, these regions of where the model is actually paying attention. So that's, it's just to show that, it's very similar types of models, but they can answer, you just change like very particular types of ingredients in these models, and you can get to very, very different types of, answering or trying to answer very different types of biological questions that are all, very important biological questions. So I'd like to leave some time for questions. But before that, I want to acknowledge, this was all of the work. I mentioned it basically when I was going through, was all in collaboration with, experimental groups and also theoretical groups. With Shiva's lab, I mentioned all of the imaging experiments were done in Shiva's lab at ETH Zurich. Then with Xiao on the tissue level data. Anthony and Stephen, I didn't mention yet, that was the heart ECG and heart MRI data. Fei Chen was the protein data. So all of these are colleagues at the Broad. And then for the theory behind overparameter, so our latent spaces are usually very large. So the theory behind that was done with Misha Belkin's lab at UCSD. Hopefully some of you guys also know him. And then with Matthias Durton, some of the causal work. And Adit was a joint PhD student with Misha's lab, is now a faculty at MIT. And Shini is now a faculty. She was doing all of the multimodal autoencoder work. She is now a faculty in Austria and Vienna. And Echachi is going to be on the faculty market. She has done all of the causal work. So she is applying for faculty positions this year. And of course, thank you for all the funding. And hopefully I was able to show that the biomedical sciences are uniquely suited not only to being one of the greatest beneficiaries of AI, but also one of the greatest sources of inspiration for it. That's where we take a lot of inspiration for when we think through, what are really methods that we need to develop that aren't there yet. Where do we need new theory? And with that, thank you all for your attention, and I'm very happy to take questions. Maybe you have to come closer. I don't hear you.
Great. So can you hear me here?
Yes, perfect. That's great.
So I think we'll pick up some questions, but I have a question first. So we have prior mouse embryo multimodal data, like we have spatial data, we have single cell RNA data, we have single cell taxi, single cell methylome. And can we use this kind of like models to correlate them with the human embryo data using the multimodal encoders that you presented? Because if yes, then we can do perturbation in the mouse, and then we can just like predict how it would be in the human embryo, because we can never do the experiment in human embryo.
Yeah. So yeah, it would be nice, no, if we could do that. So we have never trained, these joint embeddings of like human data, spatial data, and then mouse spatial data. So these are separate models, right? So I think you need something in order to be able to do some of the pairing. So I mean, it's something that we're very interested in. We're interested in the lymphoma setting, where we have a lot of human data and a lot of mouse data. But there, at least we have some pairing, where in the mouse, we did all of the most frequent mutations. So we have mouse models for all of the different mutations that appear. And then at least, there should be a little bit of pairing between them, so that I know like what structures will correspond to one and the other. But otherwise, I think you're just too different from each other that you could just directly integrate the two together. There needs to be some way of you being able to pair the two. Yeah.
I see. Thank you. Any other questions?
Yeah, I have a question.
Oh,
Sorry.
yeah, you can go
Hi,
first.
Dr. Wu. Thanks for the talk. Yeah, I have a question regarding at the beginning of the slide when you describe how you can like embed different modalities into the latent space, and then you mentioned that by adding this like contrastive loss, and then you can somehow like get the causal variables, and that part I'm like a bit confused on how like just adding the contrastive loss would help encourage causality. And
yeah, I think that's important. So why it is literally because what you're then getting out is information that is invariant to how you're looking at the system, right? So you would believe that whatever is causal should be there, whether you're looking at it from the ECG lens or from the MRI lens or from the angiogram lens, right? Whatever is causal for the heart should actually be there. It's something that doesn't depend on how you're looking at the system. And so by only looking for information that is invariant to how you're looking at it, that's what the contrastive loss does. right? It throws out all of the information that is actually not shared. We are encouraging the model to actually do that.
I see.
And the proof that this is, I mean, so there is no proof that the contrastive loss, right? The contrastive loss is just an approximation to this theoretical thing of saying like, yes, if you look for exactly what is invariant to how you're looking at the system, you actually do get causal variables out. Then one way of actually, you know, having an algorithm that tries to do that is by using a contrastive loss if you have paired data, or by using a discriminative loss if you don't have paired data. So actually the theoretical results here are even in the unpaired setting. So we are assuming that we are aligning distributions in the latent space. And so again, one way of doing that is by adding a discriminator in the latent space.
Yes. Thanks.
Or an MMD loss or something like that in the latent space.
Okay, thank you.
Yeah.
We have a question in the room. But can you come like closer here so that? Great.
Wonderful talk, Dr. Uhler. I guess my question is a bit more of a practical one. You know, say you have a bunch of these different modalities. How do you determine how do you determine which modalities are too redundant? Because, you know, you can always add more information, but like, how do you, I guess, practically determine, okay, we don't need 10 modalities, we just need like 2 or 3?
Yeah, I mean, those are very important questions. And also, you know, like people would also ask me the other thing, like, well, if I throw in a modality and it has nothing to do with the task, well, then it should somehow, you know, hurt the model, right? Or hurt the whole system. And so that's why we're so excited about this, that we now use these models where we have these partially overlapping latent spaces, where if you have a modality that actually has nothing to do with it, it's just having nothing in the shared latent space. And so we also tried this out, right? So it will just put everything into the modality specific one. And basically it doesn't change, you know, it doesn't hurt the model because you have it. Similarly, if you actually have modalities that are really very, very overlapping, like actually single cell RNA-seq and single cell ATAC-seq, they share a lot of information. There is only very certain genes, right, that like have to do with initiating regulation, say, for example, that you would see in ATAC-seq land, cell cycle stuff that you would see more in RNA-seq land. So that's when you would have a very large shared space, but very small modality-specific spaces. And so in some sense, that doesn't hurt you, right? Because now, I mean, it hurts you in terms of, okay, computation time is of course bigger for these types of models, but at least for the downstream tasks, it doesn't hurt you. And so I think that's just a great thing about these types of models.
I see. Thank you.
Yeah.
So I think we just have time for one more question in the room. Yes.
Hi, great talk. I just have questions about the image localization generation prediction project. So we know for HPA, from my observation analysis, I think about like 2/3 of the protein actually doesn't change their localization so much in different cell lines. So I think my question is like how many like proteins that you predicted actually like generated meaningful differential localizations in your different cell lines that you're predicting. And also like when you're generating your images, do you provide them like non-stained cell images to provide the cell morphology information for different cells? Because different cell has different morphology and sizes and stuff like that. And that could actually change a lot of the protein localizations as well.
Exactly.
So I'm just kind of wondering like more details about this project and what are your findings about it? Thank you.
Yeah, of course. These are all great questions. So yes, so we actually don't tell it which cell type it is, but all you're putting in is images of the cell. And as you said, and also I showed before, right, even DAPI is enough to predict which cell type it is. And so now here, all we're using are the landmark stains. So this is how we're representing a cell. Okay, so the landmark stains of a cell go in. And then based on these and, you know, whichever protein you want to produce a localization for, the protein sequence goes in. And then out comes the image of how that protein would look like in this cell when you were to stain for that protein. Okay, so that's how this works. And so now you can generate, and of course, if you were analyzing, I'm assuming you're analyzing the human protein atlas where you're looking at, localization of particular proteins across cell types. Of course, you don't have every protein across all cell types, so you can actually not do the analysis of saying which proteins are really varying across cell types because you don't have the data, right? So we were able to fill in the full matrix. So you only have 0.25% of all cell line protein pairs. So because we're generating it, we could actually fill it up to 100%. And then we can really answer the question of, which proteins are varying localization across cell types, but also which proteins are varying their localization within a cell type. Okay, for sometimes actually even within a cell type, you have variability across cells. And so those are the types of questions you can only answer if you can actually generate, if you have a computational model to generate this.
I see.
Yeah,
Thank you.
and so as you said, we can make changes in the landmark state. So you can put in a new cell, like what we did, right, with a completely new cell type that is actually not even contained in Human Protein Atlas, and try to predict where all proteins are for that cell type, or for that cell. Or you can now change the protein sequence. So I can make mutations in here and see how that changes the localization.
Thank you so much.
Yeah, thank you. Those are great questions.
Thank you. So do you have time for one more question on the Zoom?
Sure.
Or yeah. So Nathan on the Zoom.
All right. Caroline, hi. Great representation. Truly enjoyed it. My question was still again like the joint latest space that you are creating with different modalities. One of the things that we are often particularly worried with, especially like in multiomics and other multidimensional technologies, is batching, right? Is batch effect and the reproducibility of our results across different experiments. How have you explored this in your with your methodology? Like, is the latent space that you explore in one experiment translatable to a different experiment?
Yes. And so that's also where, so since we proved that you can learn causal information from multimodality, batch effect is not causal, right? So the first thing we always show is that batch effect is gone when you have multimodal data. And the intuition is, again, is like, I mean, maybe simple once you hear it, right? Because the batch effect that you have in these images, like say, if we do it on spatial transcriptomics, that you have in the spatial transcriptomics readouts, or that you have in the Hurst staining, they're different batch effects. And so if I'm looking for what is invariant to, the different modalities, you're actually directly getting rid of batch effects. And so in a paper where we have a lot of H&E data and spatial transcriptomics, there again, we were able to actually integrate even studies that, now this was in ovarian cancer. And so we were like, there was just a new study that came out of Australia, a very big ovarian cancer data set. And we're like, wow, okay, this is great. So let's try to integrate it. And those are, of course, huge batch effects. But because we're looking for what is invariant in their study between their H&E and their spatial transcriptomics, and what is invariant in our study between H&E and spatial, we were just able to directly integrate it. No batch effects in the latent space. I mean, you cannot identify anymore whether you were in the data set from Australia or in ours from the Broad.
Yeah, that's fascinating. So basically, your latent space is not really like a representation of correlation between these two different modalities, because this correlation could easily arise because of batching, right?
Yes. So no. So you're looking for information that is invariant to it. So now there are modalities where we saw that this type of thing doesn't work, where the batch effects seem to be similar. So if you have many different protein stains in the same image, right? So it's just different, the color channels are different proteins. There it seems like the batch effects are just the same, independent of modality. And so the more different the modalities are, the better it is to do this. Like your ECG MRI stuff, right? The batch effects will definitely be different, or our H&E and spatial transcriptomics. That's where it really helps to do this, to get rid of batch effects.
Lovely.
Yeah.
Thank you.
Yeah, great questions.
Hey, thank you. Thank you for being here virtually today.
Yeah, well, thank you for having me.
Yeah.